Genetic mutations and DNA repair.
Mutations, they are one of the reasons for genetic variation in organisms. They can be inherited and there are many types. As listed here, these are examples of some mutations. Let's start off with some examples. Now, this is a transition. Transitions are when there's a base mutation between two purines or two pyrimidines. However, transversions would be a base mutation where purine is substituted with a pyrimidine. After showing some examples where there is a base substitution, they can also be inserted or deleted. But first, we're going to show you a basic base substitution. And so here is the original DNA sequence. The original DNA sequence is shown above. Now this is a base substitution. Insertions or deletions can cause the frame to shift. Rather than changing the nucleotide, an insertion will insert a new nucleotide into the sequence. On the other hand, a deletion will remove a nucleotide from the sequence. Both insertion and deletion will end up causing a frame shift, and these frame shifts ultimately affect transcription and translation. Now, that brings us to functional mutations. There are effects caused by the mutations shown before. There's forward, reverse, missense, nonsense, and silent mutations. Now, let's get to some examples. Here's an example of no mutation, nothing to enrich his life. They would transcribe and translate normally and end up producing a wild-type protein. Just another day at work. Now, let's show missense mutations. Missense occurs when there is a change in the codon from the DNA strand, so the mRNA transcribes incorrectly and ultimately makes a different protein. It's like if someone put in the wrong fruit order and end up getting apples instead of strawberries. For nonsense mutations, there is a single nucleotide base change where the codon will encode for a stop codon instead of a protein, so it prematurely stops the translation process of the amino acid chain. Missense and nonsense are similar, but you can think of nonsense as making no sense if the amino acid chain has no more proteins encoded after the mutation. Missense is a mistranslation, so it's mistakenly made another amino acid, different from the wild type. Finally, there's a silent mutation. Silent mutations are mutations that have no change to the phenotype of the amino acid. Amino acids are more than one codon that can encode it. Thus, there can be a single base mutation as seen in an example, but the translation process will produce the wild-type protein as if there were no mutation.
Mutations may be caused by a number of different factors. Spontaneous mutations are mutations that arise from natural changes in DNA or from errors in replication. Spontaneous replication errors include tautomic shifts, mispairing due to other structures that often arise through wobbling, incorporated errors and replicated errors, and replication and crossing over deletions and insertions. Spontaneous chemical changes include depurination or the loss of a purine base from a nucleotide, and deamination, the loss of an amino group from its base. Induced mutations are mutations that result from environmental agents such as chemicals or radiation. Chemically induced mutations are caused by mutagens, base analogs, alkylating agents, deaminating chemicals, hydroxylamine, oxidative radicals, and intercalating agents.
There are two main categories of mutations, somatic and germline. Somatic mutations happen in non-reproductive cells. Here's an example of a mutant cell. This mutation can be passed to new cells by mitosis, creating a clone of cells with the mutant gene. Here's a population of mutant cells. With germline tissue, germline mutations can happen in cells that give rise to gametes. This is an example of a mutant cell. So meiosis in sexual reproduction allows germline mutations to pass to about half of the next generation. This fish has all its cells carrying the mutation, and this fish has no cells carrying the mutation.
Transposable elements, also named transposons, are DNA sequences that can move about in the genome and cause mutations. There are two forms, replicative and non-replicative. Transposons will pick an area in both strands of DNA, causing a break leading to gaps in both strands. The transposons insert themselves in the strands, leaving gaps open where the DNA polymerase can come in and refill the gaps with the appropriate nucleotides.